Hi, my name is Nathan Lazar, and I'm a PhD candidate at Oregon Health and Science University. And I'm going to be talking a little bit today about my dissertation work, which is building a new computational model to predict the response of cancer cell lines to drug treatment. So this is in the general area of precision medicine, where we're trying to use the genetic makeup of a patient to better inform the decisions in which drug we should use to treat that patient in order to get better responses all around. But of course we can't treat every patient with every drug and see which one they respond best to. So how can we get at this problem? One approach that researchers have taken is to, instead of treating the patient directly, we derive a cell line from the tumor cells and treat that in the lab and see how the cell lines respond to different drug treatments, which then informs how to treat the patient. And can also be used to discover relationships between the cell line genomics and the drug structural features. So we have these two problems: drug recommendation, we're predicting response, and relationship discovery. But what do we mean by cell line response here? What's normally meant by response is one summary value for each cell line drug combination. Those summary values are derived from this type of dose-response curve, where on the y-axis you have some measure of the cell line growth. On the x-axis you have increasing dosages of the drug. So as you increase the dosage, you tend to see normally that the cell line grows less in presence of stronger drug. And then you can extrapolate one value, say, from that dose-response curve. Either IC50, EC50, which is kind of halfway values, or area of the curve are all commonly used measures. But what this is doing is really throwing away a huge amount of data. We actually have responses at each dose, and instead we're boiling that down to just one number. So my idea is, instead of trying to just get one summary measure for the curve, why don't we find a way to predict the whole dose-response curve for a given cell line drug combination? This is a type of multitask learning where we're trying to predict several values for each of our inputs. And our inputs here are the genomics of the cell line and the target and structural information from each of the compounds. So how are we going to make these predictions? Well, we start just by holding all the data together in one three-dimensional matrix or tensor of numbers. So for every cell line, compound, and dose, we have one measure of response. And as the doses increase, the growth goes down, or in this case, it's more blue. So we take this three-dimensional tensor and then break it apart into latent representations for each of those dimensions that are used to predict the response. So we have a latent dimension for cell lines, for drugs, and for doses. And for those dimensions where we have some type of predictor data, that latent representation is required to be the product of our input information and some sparse projection matrix. So the projection matrix is sparse row-wise, meaning that most of the values here are 0. And if a row is not 0, one row here corresponding to one piece of information, be it gene expression value or copy number data, that's useful in predicting a response for that cell line. So the algorithm learns these distributions on these projection matrices and can then predict by just multiplying a new cell line with the input information by this projection matrix, forming the latent representation in order to form the response data. So I can have, for a new cell line, for instance, I can get drug response curves for my whole suite of drugs. For a new drug, I can get curves for all the cell lines that I've measured, or I could even do both at the same time, have a new cell line, a new drug, and get some dose response curve prediction from the algorithm. So this algorithm is being trained using the largest data set of this type that's been generated yet from the CT squared effort out of the Broad Institute. And I'm training it using this method called variational approximation, which is much more efficient than Monte Carlo methods, and actually learns the distributions on each one of these values instead of just making a point estimate. So it gives us some measure of uncertainty when making these predictions. So I'm looking forward to talking about this more at the upcoming meeting, and I'd just like to thank my committee and advisors and my funding source, National Library of Medicine. Thank you.